Hello, welcome back to COG 266, Brains, Minds, and Consciousness. Today we are going to continue our journey sort of up through the brain. We've been focused very heavily on individual neurons and how they work and how they communicate. And now it's time for us to kind of catch the wind and move upward a bit so that we can get a little bit higher up view of how the brain does what it does. So to do that, we're going to take one function of our brain and mind, vision, one of our senses, and we're going to follow it upward and sort of see as it gets more and more abstract and more processed how these different processes work. So, to recap, we have discussed 5 of our senses. You actually have others like vestibular sense, like balance, and other kinesthetic senses and stuff. But these are the 5 main ones that we all know. You know, you're taught in school, right? We have touch. We've talked about how touch mechanoreceptors work. How the deformation of the membrane will cause ions to flow into the cell and cause a charge, cause a signal to be propagated. We've talked about hearing, how the basilar membrane vibrates and makes the hairs on the receptors in there move at different frequencies. We've talked about taste, how different molecules of food can bind to taste receptors to cause signals to happen. We've talked about smell, how similar to taste, things in your nasal cavity will catch. You have hundreds of different individual receptors in the nasal cavity, in the sinuses to catch these smell particles. And sight, how light will hit the receptors, hit proteins in the receptors of your eyes and cause, a chain reaction of other proteins to move off and cause changes in cells to make signals happen. So, all together, these are the receptors that we have discussed, that we have shown more or less how things out there in the world cause these receptors to trigger and send signals onward. Hey, now we can get signals from the outside into the brain. That's pretty cool. So, again, now our goal is to follow one of these senses. And we're going to go from the bottom up. So we understand how, an individual neuron can propagate a signal and how it can communicate to other nearby neurons. And we also kind of understand how these sensory receptors can get information from the outside world into our neural system. So now the goal is to follow how one of these senses moves upward. So we get the basic signal in. Cool. How do we turn that into a complex thought, right? So from the bottom up. Today we're going to follow vision. So vision is the best understood sense, and that's because it's easier to research than a lot of things, because there's a lot of your brain is dedicated to processing specifically vision, and it does so in a very sort of predictable way, and it's also easy to change visual stimuli in the world and study what happens in vision, not just in humans but in apes and other animals. We can study vision from a lot of different angles because it's very easy for us to change light patterns. A little harder to study things like smell. It's a lot slower, more complex, right? Not to say vision is not complex. You'll see how that works soon. But here's the thing. Early vision. So as we begin the journey deep into the brain to say, hey, here's a bunch of symbols on this screen. They mean things like the words early and vision. That is something that your brain is handling down the line, but it starts as just photons hitting your eye receptors, and then something must be processed later on. So after the pattern of light hits your retina, it's converted into this array of visual signals, a whole set of signals all together next to each other. But the trick is it almost immediately begins getting processed. We can look at this, how this sort of came about, how we uncovered this interesting tidbit, by looking at the horseshoe crab. So, horseshoe crabs are these strange-looking critters. They are little, I mean, they're crabs, but they're almost more primordial than that. They are these insect-like crab creatures. They have eyes that are sort of, again, insect-like in nature. They have these sort of outward-facing detectors, right? So you can see the eye at the bottom here. It's very hard to see, but we'll talk in a second. They are faceted, kind of like a fly's eye. You know, with a classic look at a fly's eye, it's this, you know, many sort of domed shapes. It looks like a lot of flat little surfaces, right? That's because that's what it is, essentially. So horseshoe crabs, they have two prominent eyes on the surface of their domed shell, and they have about 1,200 facets per eye. A facet just means like a flat surface on what is more of a rounded overall structure. So there's about 1,200 facets on the eye, and in each of those is an ommatidia, which contains about 15 different photoreceptor cells and a single eccentric cell that is going to catch all of the light from the various photoreceptors. So there's a bunch of photoreceptors you can see on the bottom right here, but the lens on the outside, the part that is actually taking the light in, so the light comes in there, the photoreceptors catch the signals as we've discussed, and then the eccentric cell does something and passes the signal on. The question is, what is that something? What are we doing at this stage? So, basics of observing these ommatidia in action, right? So we can observe them, the reactions to patterns of light, and the patterns of their reactions as well, using what are called single cell recordings. We can actually pinpoint an individual neural cell, especially in these horseshoe crabs, because they have nice big neurons. They're easily to separate. And so the basic idea is that at first they worked sort of in the basic sense of how we would expect them to, right? More light increases the overall response. And the assumption is that, okay, so each one of these little receptors, they are detecting light, and they are able to take that light and send it on to the internal brain to get processed, right? The raw signals just come in and we send the information onward. Now, that is a perfectly happy explanation to say, okay, I'm getting light on each of these little facets of my eyeball, and I'm sending that into my brain for processing later. But there's a curiosity here. Some of the signals are actually dampened if the lights in the room are left on. That's odd, because you would expect that if the lights in the room being on, that if they were on, then all of the neurons would then be more active, right? They would have a stronger signal, but that's not the case. So, and again, not this carefully controlled stimulus patterns, but just overall ambient illumination. What's going on here? So for an individual ommatidia, the light pattern, so the response pattern is what you might expect. When the light is dim, you get a little bit of pulse. When the light is more, you get more moderate pulsing. And then when the light is very bright, the neuron goes, whoa, 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 boom, 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 right? But here's the trick. If I'm looking at multiple of them, then, so X, Y, and Z, these separate different ones, so when there's a lot of light going in to multiple adjacent ommatidia, then their overall pattern of activation is lower. The pattern of activation of the X, the X ommatidia is lower when Y is lit up. And then when Z is added, it sort of comes back. But that's interesting. So what good is that? If one ommatidia is active, it behaves as expected. Light makes more activation. But if multiple adjacent ommatidia are active, they laterally inhibit one another, such that if there is bright light right next to dim light, there's this incongruous drop-off in activation of the given ommatidia. What is that about? Why would you need to do that? Why would that happen? But, I mean, on top of that, the more basic curiosity, the more basic takeaway from these studies is the signals from each light-detecting cluster in the horseshoe crab are all wired up with each other. The information is being processed before it ever even leaves the eye. So it doesn't need to go to central. It doesn't need to go all the way to the brain to start changing its shape. It starts doing that the instant it leaves the receptor. And in fact, in a way, as it leaves the receptor. That's interesting. What's it all about? What good would this kind of processing do? Well, let's talk about ganglion cells in your eye. So, Hubel and Weissel, 1959, they started looking at ganglion cells in the eye. So ganglia respond to light in a particular region of the eye. These are sort of, if you think about them as they are the neuron that catches several rods or cone receptors all at once. So they have a reduced response to dark in the same region, right? So bright light produces a certain kind of response. Dark light, a lack of light, a darker light shows a reduced response, right? Here's the tricky part. As light leaves the ganglia's region, that pattern reverses and it starts responding again. So you follow that? There's the most activation when there's like brightness in the center of the cell. There is very dark very little activation when the light is at the edge of the ganglion's region. And then when the light is outside of the ganglion's region, suddenly there's activation again. Not as much as when the light is on in the center, but it comes back. It has this sort of inverted, this like shape to it, right? So what this is called is center surround cells. Ganglia have a concentric receptive field, on-off center surround. Okay, what does this mean? So there's this pattern of excitatory and inhibitory connections. So there's all these negative connections in the outside concentric circle. There's a lot of positive connections in the inner circle, and then outside of that is just neutral. So the idea is that you have this pattern that's able to tell not just, hey, there's light. It can also say, hey, there's light in this specific edge spot. Ooh, how does that work? So we'll get there. So the retina is like covered with loads of these. There's overlapping center surround ganglia all over the place. This is, so the first layer is all the rods and cones, and then one layer removed from that is the ganglion cells. So what we should be getting here is that it's not just a one-to-one, hey, light is here, light is present, so I'm going to light up very brightly. It's not like a computer monitor, right, where just every pixel has to be exactly one brightness of 3 different colors, and then we're good to go, right? What's actually happening is that we begin processing immediately in this way that's able to detect not just light is present, but also differences in light are present. When I have negative weights on the outside and positive weights in the middle, that means that I'm very excited by things like bright circles in the middle with dark borders around them, and I'm very not excited by the opposite of that. So a bright light on the surround and then a dark shade in the middle. So what good is this? It's a little bit fuzzy to start, but we can talk about how we can use this for something really important. Consider this edge. This is pretend it's the side of a cube, right? The right side is sort of the darker, the shaded side, and the left side is the front face of a cube. Being able to imagine this and detect this in your eyes, it's pretty useful to have a set of things like ganglia, that are able to be really excited by differences like this. The way that we can pull in this concept of an edge, as in a difference between two colors, right? So what we're seeing right now is there's this cream color on the left and this sort of grayish color on the right. They're not that different in luminance. They're not that different in terms of their interest, but they're these two kind of murky, just light patterns. And the fact that the center of this stands out so much to your eye, it is a construct. It is a concept of a line. There's no line there. It's just two differences in light. You create the line internally. How? The ganglion activation. So, as an edge, and we can do this with what we've already discussed, right? As the edge passes over the center of the ganglion, it changes how active the cell is. So to start, if one particular ganglion is just completely inside of the bright area, right? You'll get some negative activation from the surrounding cells, and the center cells will get plenty of normal activation. You get sort of an evened out, low-key activation. However, as the dark area starts to pass over the edge, we end up with this interesting boost, because now there's a whole lot of positive connections to bright light in the center, but some of the outside has been inverted with the darkness. So the darkness hits the outside edge while the light is still on the inside center areas. When we sum all that together, remember, we're summing activations like we've talked about before with neurons, you get darkness on the edge means this cell is really excited because these negative weights are pairing with the positive weights with the light from the center, and we get lots of activation. Then it starts to drop off again because the negative weights for the darkness start happening. The positive area, the center area that likes bright light, is now getting darkness. So it sort of tapers off until the whole thing is now covered pretty much in darkness with only a little bit of positive weight, and now we get almost no activation. But again, very different from the first. So on the far left, that's kind of the normal activation of, hey, there's brightness happening, my cell's going to be active to a certain degree. When there is an edge overlapping the surrounds, this cell lights up or goes very dark. Either way, that's an important difference for us to be able to use. This means the ganglia are excited by edges. Now, we're excited. We have this individual ganglion cell that is able to be pretty excited when there's an edge in the visual field. Now, pair a few ganglion cells together, say in these patterns, you can have a new cell a couple layers back that's weighted by, hey, I only want to be excited when the vertical row of ganglia are active and not when the other ones are. I only care about a vertical edge or a diagonal edge. So now you're starting to see how not only can we detect differences and edges, but now we can detect orientation. We can bring in this concept of there's a diagonal line in front of me. If you think about how that's useful in the real world, a lot of horizontal lines in the world, good, a lot of vertical lines in the world, and being able to detect the difference between, hey, there's trees on either side of me, and hey, that's a cliff edge that I might not want to walk over, that's pretty important. These are useful cues that we can pull in from the outside world and not just have it be this raw dot pattern of light, but we're actually getting into constructs, into things that are useful for us. So these groups of adjacent ganglia can give us this detection of orientation, and then further up in the brain, they can be tuned to specific set of ganglia for these different orientation responses. So that's really great. This is how I hope you're starting to see how we can go from, hey, there's some light in this position, to, hey, there's differences of light all lined up in a particular way that gives me something pretty important to understand. There's a tree over there. I might want to investigate it or avoid it if I'm afraid of things falling out of trees. I don't know. I don't know what environment I'm coming up in this world, but that's the idea. So these basic shapes you can continue to abstract further and further until you get more complex things. Like, I only care when there are a certain number of diagonal shapes next to each other forming, say, a face, right? So that's the idea. I hope it's making sense how we go from single receptors up into more abstract, complex structures. And yeah, I mean, this is a very cool thing. And this is all before it even leaves the eyeball. You know, it is on the way into the brain. It's already getting chopped into these various kinds of structures. These new, more complex pieces of data that we need to get from the outside world. Now, another thing, an interesting tidbit about how these work, we're talking about how the outside edges are more are happier to see darkness, and the inside is happier to see brightness. This causes a funny little thing. Mach bands. So if you look at this array of vertical rectangles of varying grayscale, you might start to notice a little bit of a funny feeling that there's a little bit more darkness towards the edge. So so each individual vertical stripe here, it looks like it's kind of different colors, right? You sort of see a gradient to it. Each one is a little bit lighter on the left side and a little bit darker on the right side, right? That is not the case. In fact, that's not true at all. These are solid colors. I haven't shaded them in at all. The funny bit is that because your system is, because of the way your ganglia work, you are seeing they're a little bit more excited when there is that difference there. So things look a little bit darker or lighter depending on that difference. This is how we think edges are more important. Or this is an example of one of the interesting little artifacts of the fact that our brain really wants to detect these edges. It makes it kind of blurs the reality a little bit. Similar effect with these center surround scenarios. So if I asked you which one of these had the lightest center, you would absolutely say the left one, right? But that's not the case. They are all the same luminance. The centers are all the same. The right one looks darker, the left one looks lighter. There's a difference effect going on here. We are seeing that certain areas are being more lit up or more darkened or whatever, more active, influences the way we experience the other ones, right? So this is not maybe at the linear level. It's not about this detection of a line at these positions, but it's more about something further up the chain is causing a difference of, hey, there's a lot of dark going on here and this part is brighter. We should probably think of that as brighter. And that's useful. In the real world, things do kind of work like that. But we can kind of mess with our visual systems. This is what all the optical illusions are about. You can mess with your visual system by doing these little artificial kind of fake things. So, all to say, it sounds like these things are fake, they're illusions, whatever, but honestly, these are more real than anything else because this is how your eye sees the world. You know, it's not fake. It's that your version of things that comes into your visual system, that is darker. That is lighter in these effects. Because in the real world, things that are like this, it actually kind of, I do need to consider that as darker. I don't care that it's the literal exact same luminance. It's all about differences, relationships, connections between things and their surroundings that are the most important. So, all this to say, this is all happening, for the sake of, it's not like, quote, all in your head, it's literally all in your eyes in this case. Here's another illusion I like to show. This one's interesting. Look at any given intersection and kind of move your attention without moving your eye. Just keep your eye fixed on one of these intersections, the little white dots. The other intersections further out from your center of vision, they kind of look like they have black circles in the middle of them, don't they? But move your eyes and you suddenly detect, no, there are no black circles. There's no black circles at all. And yet, this is showing how your ganglia will really, really tell you, like, hey, there's a difference over here. This is probably what we're seeing. The reason this is kind of showing how your cones in the periphery kind of work a little differently than the ones your rods in the periphery work a little differently. The sort of the ganglion cells in charge of integrating the rods is getting a different kind of signal than the ones in charge of integrating your cones that are in the center. All is to say that this is one of those ways that we can construct a precariously, very precisely tuned way to trick your eyes into seeing something that's not actually there. It's fun. It's interesting, right? And I think you should hang on to these experiences just to remind yourself of, whoa, my eyes are really more complex than they might seem. But ultimately, this kind of stuff doesn't really come up in the real world. That's why our eyes have evolved the way they have. It's really kind of useful to be able to be tricked in this particular way, because the way things actually work, this won't come up, right? So, all this to say, it's fun. It's interesting. Your vision system is very interesting. It is very complex, and we've only kind of scratched the surface, but we're not going to stay forever on these topics of how these things all get integrated up, because that's not the focus of the course. So, early vision. Before the information even leaves the eye, the ganglia start processing it into these meaningful edges, right? And these center surround cells help us detect adjacent differences, which then, going up, can be combined in similar ways by later neurons into larger structures, edges, surfaces, more complex shapes like faces, like eyes, like teeth. A lot of things, on faces. Human social stuff is pretty important to our brains, but yeah. So, that's early vision. This is how shapes start to take form as they move from light patterns into our brains. Just a little stripy show as we move forward away from just shapes, away from just grayscale, into color vision, the second unit of this lecture. So, color vision. We are able to see color. We know that light is this electromagnetic radiation that disrupts our receptors in our eyes and lets them fire in different ways, different patterns, right? White light comes into a prism and is split off into all these various colors, right? The idea is that there exist many different wavelengths of light that are all kind of bouncing around in true white light, in the kind of light that we get from the sun, the kind of light that we get that's just sort of a blend of all other kinds of light. We get it all, and it's all sort of out there, available to be parsed. There are two major properties of light that we use to detect what is going on in the outside world, and getting color, not just shape. So there are two major physical properties of light. There's the wavelength. So if as white light behaves like a wave, it also behaves like a particle. That's quantum physics, so we're not going to get into that. But as light often behaves like a wave, that means it has the properties of waves. So it has amplitude, so how far up and down, how wide the wave is, and then the wavelength of how frequently it moves up and down, right? So a very low wavelength means a very high frequency. It's gonna wobble a lot, right? Whereas amplitude means how far does it wobble. Wavelength means how fast does it wobble. I know it's a little much. Just remember this diagram of like wavelength is sort of how compressed it looks, and amplitude is how tall it is. Altogether, the point is that these are two physical properties of light that we can use. So we can extract interesting information like brightness, hue, as in the color, saturation, as in how much of the color is there versus how faded out is it. So let's talk wavelengths first. Different wavelengths will cause different experiences of color. Red light's wavelength is about 700 nanometers, green light is about 530 nanometers, and blue light is at about 400 nanometers. These are also the sensitivities that our cones are tuned to, right? White light is sort of a blend of all of these. It's not one particular wavelength. It's a boatload of kind of random wavelengths out there in the world. So green light, our preferred green light, is this 530 nanometers, but we can still detect all the lights near it. So 520, 540 nanometers, we can still detect all of that. But ultimately, we have these three kinds of wavelengths that we are tuned to. Reflectance, so this is kind of important to get at the base level. The wavelengths of light that are reflected off of a surface and not absorbed by it, that represents the color that we actually are able to see. Now that makes sense now that we've talked about this, but it's a little weird to think about how, the leaves are green, meaning they reflect green light and they absorb all the rest. You kind of don't think of it that way, right? Or it seems odd to think of it that way, but that is the ground truth about reflectance. Now, the amplitude of the light bouncing off is also going to reflect the brightness. So higher amplitude means higher brightness. So amplitude has to do a lot with the energy of the light. There's a lot of motion going on. So most light is not monochromatic, but it's that mixture of lots of wavelengths. There's a lot of light going on. So, for example, ground beef has a certain pattern of reflectance, right? So it's going to reflect light of all kinds of different wavelengths across the spectrum. A little bit more of the longer wavelengths, so a little bit more red, right? Whereas there's a little bit less of the other ones. However, when you look at the profile of cooked beef in the same stuff, same chemical stuff, same properties, same entity, but later then cooked, you end up with a little bit more of these sort of higher end wavelengths and a little bit less of the lower end ones. You end up with a different profile of reflectance. You get more of this brown with shiny spots as opposed to this sort of matte red across the board. That's very important. This signals something. Even though it's the same object, it has changed in ways that are very important to know the difference about, right? You don't want to just eat raw meat. That's not great. Everyone talks about rare versus whatever. You don't want to eat raw ground beef, I promise you. But cooked has a different set of properties. So these are ways that there's complex information available in the way that a whole set of light wavelengths are coming off of a particular object. Now, let's talk about light mixture. There are two ways of mixing light. Now, this is some physics 001 stuff, but two ways. If you remember in grade school, you might have mixed paints together. That's subtractive. So you add substances that will absorb more wavelengths of light. So if I mix the yellow paint and the blue paint, then each of those pigments is going to absorb its own field of wavelengths. So the yellow is going to absorb most things that aren't yellow, blue is going to absorb most things that aren't blue, and they're each going to absorb each other's until we are left with green, right? So that's how mixing paints works. You are taking away by absorbing more of what's happening. That's why black paint absorbs almost all the light, and honestly gets quite hot. If you have a black car, that's why it's hotter in the summer. It absorbs more of the light. Now, additive light mixture has to do with adding additional wavelengths to produce a new combination of wavelengths. We typically detect sort of the average wavelength of things. So shining multiple lights is additive. So each light is going to add more wavelengths, and we combine to get the sort of average. Now, interesting tidbit. Metamers and non-spectral colors. Metamers are light experiences of light, right? So our understanding of the concept of orange is it can be represented two completely different ways. We can have one perfectly, one single wavelength of light shining somewhere. So just like from an LED, a perfectly tuned orange LED. It is the identical experience to the experience of summing up two different lights that are near that frequency. So 580 versus 650 added together. It's non-linear. It's a little odd. I don't know all the math, so just trust these things. You combining two different wavelengths, you get the exact same experience of orange. Whereas purple is actually a non-spectral color, which is odd, but it kind of makes sense, right? Blue is very high frequency, red is very low. Combining blue and red gets you purple. There is no raw single color that gives us purple, and yet we're able to experience it strictly through the combination of having lots of blue happening and having lots of red happening. That means it's not really there on the spectrum. It's sort of this construct. Now that's just sort of food for thought. It's interesting. It's a fascinating little tidbit that there are light frequencies that we sort of confabulate versus light frequencies that are extant in the world. How does that all work? Well, the trichromatic receptor theory is the idea that every color of our perceivable light can be produced by a combination of three colors of light. The reason that exists, the reason that's possible, is because that's what our cones are tuned toward. So in the bottom left, we have a lot of red light coming off. That activates our red cones. In the middle, we have blue and red activating together because lots of blue light is coming out and lots of red light is coming out. That gives us purple. We just combine them internally in our brains, in our eyes. Yellow, similar thing. We have lots of red and green coming off, almost no blue. We get yellow. This is why your monitors on all your devices typically have three colors available. It used to be just red, green, and blue. Now it's, magenta, cyan, and yellow. But the idea is that still we can trigger these receptors in our eyes using a combination of three lights. So, color cones. The trick with these is that each one, they don't just accept one color. They accept a band of colors, and they are highly tuned towards different ones, right? So the red receptors get a lot more of the high-end reddish, like longer wavelength colors. The green receptors focus on all these greens, and that's kind of important because so much of our world is green in nature. And then blue receptors kind of focus on the low end. These are called curves. These are graded, right? So as we saw with the ganglion cells, your rods and your cones will, if I'm giving you a very, very, very dark blue, the red and the green ones are not going to activate much at all, almost none. The blue one will activate a little, right? So this amount of blue, we are able to say, oh, that's a dark, kind of a dark bluish, right? But if I receive green, well, that's going to activate almost all of my receptors, sort of this like, right where the word green is there, roundabouts that area. that actually will activate all of the different neural receptors, the different color cone receptors, but green will be most lit up. Red will be lit up a bit, and blue will be lit up slightly less than red at that particular vertical. That's the idea. I hope it's coming across. I hope you're getting that your cones, they are actually receptive to more than just one exact color. And in fact, it would be very hard if they were only receptive to one color, because that would make it very hard to detect anything in the world at all, because what if that color was absent? So this leads us to the concept of color blindness. Those who are colorblind aren't just, oh, everything's grayscale, right? And it's not just that, oh, they can't see green or red. No, it's more that one of their cones is not really developed. One of their cone types, right? So normal vision, you get the full rainbow. Deuter anomaly, you get sort of this dampened rainbow, proto-anomaly. There are all these different kinds. Some people have absolutely no different cones. They have one kind of cone, so that all they can get is brightness, right? So that's achromatopsia. That's the it's not especially common. Deuter anomaly is the most common, and then the others all have different amounts and different varying amounts of like what is and is not available. If we look back at this tuning curve, imagine removing one of these curves. That's how you get these different profiles of color blindness. So, real quick, in case you haven't known, you might be colorblind. Can you see the number here? It's 8. Can you see the number here? It's 5. Can you see the number here? It's 42. Now, I'm not going to harp on this, and sorry if I just blew your mind. Maybe you didn't know you were colorblind. If you are, it's okay. Don't worry about it. That's something that you might want to know, and you might look into. But ultimately, a lot of things can be designed with your color vision in mind, and with your lack of color vision to some degree in mind as well. And that's what these tools are for, right? That's what these patterns are developed to help figure out if someone is lacking a certain amount of these cones or not. It's all good. So, you're not alone if you are colorblind, because we all are colorblind in different ways. Because if we think about our human eyes, they do this great job. They get 3 different colors. Very cool. Fine. Yeah. We're not the only ones that can have special receptors, though. So, whereas dogs might have fewer colors available to them, so the color is coarser and less defined, some animals have a bunch more colors available to them. Butterflies and mantis shrimp have a lot more types of light sensors, meaning that they can detect things that we literally cannot imagine. As in, they can combine various sources of colors in the ways that we've discussed, which I hope makes sense already, to get access to a rich color profile of the world, a complex experience of seeing colors reflecting off of objects. We can't even picture those colors. That's not possible for us because we've never been exposed to them. We don't have the neural receptors that we would need to experience those colors. So we don't. We are all colorblind compared to the mantis shrimp. Important takeaway. So, what's the fun part, right? These all have some kind of interesting anomaly we can mess around with. So there's something interesting here. The cells that we have that allow us to perceive color, they are actually kind of lumped together. So opponent process theory is this idea that an increase in our perception of red causes a decrease in our perception of green. And the opposite is true. So when there's a strong green response, these receptors go dark, which is fine. Again, remember that whenever we talk about a cell going dark, that doesn't mean that it's no longer useful. In fact, a complete lack of signal can be just as strong as a very strong signal. Remember, we can always invert signals in the brain. You can have an inhibitory connection versus an excitatory connection. So the point is that there is a strong difference, right? Red means very far in one direction, and green means very far in the other direction. The opponent process theory. And what this causes is a very interesting phenomenon. When a color goes suddenly absent, the cells kind of overcorrect a little bit. This strong difference suddenly happens, and we detect the opposite color. So, for a moment, stare at this pattern. I am also staring. I'm trying to induce this in myself. I'm looking right at the center. Just keep your eyes fixed, because otherwise you're going to be refreshing all of your opponent processes over and over, so keep them in the center. And let your eyes get really used to this image of a flag. And then, all of a sudden, it will change. Now, if you blink a little bit or if you move your eyes too much, it'll fade away. But for a moment there, I hope you detected there was a flag, a familiar flag, and then it vanished. So that's interesting. You get these impressions, these after images. Also, if this didn't work for you, I encourage you to try it again. Go back and pause the video and try it out. We'll see if it works. But the point is that your eyes are constantly trying to sort of tug of war one light color versus the other. So we might have 3 distinct photoreceptors, but right there at the level of the ganglion cells, they're already being reprocessed into a different form. Again, similar to the shapes, we're also getting that in color, right? So it's not just, hey, send on the blue, red, and green. It's saying, send on the red-green, the red versus green, send on the blue versus yellow, and send on the dark versus light. You could maybe imagine how you might hook up these different cells to these different elements to sort of give inhibitory versus excitatory connections, whatever. But the idea here is that you can recombine those 3 raw color signals and turn them into something that apparently is more useful. Now, I wouldn't have known to say, hey, red and green, those should be opposite. Blue and yellow, those should be opposite. I wouldn't have figured that out if I were designing an eye from the start, but this is what works for us. These are the color oppositions that work for us. And also, ultimately, we can still detect all these different colors. So whatever form it takes on its way in isn't hypercritical. Okay, so color vision. Eyes are sensitive to colors based on the wavelengths present in them and also the amplitudes. We combine information from detectors for each wavelength to get the picture of color in the world. Sometimes that can be impaired if we're lacking a certain kind of cone. But there's also these processes that cause a sort of an opponent, a push and pull version of this. It's not just sending light straight in, it's sending in sort of these more densely compacted bits of information about, hey, how much green versus red is present. And it causes this interesting effect of after images. So complementary colors will appear in response to them vanishing. So that's vision part 1. I hope you're starting to see here how both shapes and colors can be the result of combined patterns of light in the world. So shapes come from adjacent differences, which can be transformed into edges by connecting a bunch of neurons together, which can be transformed into surfaces by connecting a bunch of those neurons together, right? So as we propagate upward, we start to detect different types of shapes. Same with the light. The 3 different wavelengths come in, they hit the ganglia, and then we extract things like hue and brightness and these opponent processes. Next time, we'll follow how these basic elements get transformed and quantified and abstracted upward as we continue on into the rest of the brain. As always, a final thought. Consider the fact that visual information starts getting processed into new forms before it even leaves the eye. Is this the case with other senses? What about in other places distributed throughout your body? How might these answers interact with the common sense conception that cognition happens in the brain? All right. That's it. Thanks. Take care.